Você quer que eu fale uma coisa? Eu falo também. Bom, pessoal, obrigado por terem vindo. Agradeço a presença de todos. Agradeço também a presença de todo mundo que está assistindo no YouTube. A gente teve um mês muito interessante com 2 convidados internacionais, né, em setembro. E isso, eu queria fazer parte, né, está associado com a programação da SBNEC, da Sociedade Brasileira de Neurociências, o congresso que aconteceu semana passada, e que é organizado em parte pela professora Débora, e os convites, né, feitos pelos docentes, os pesquisadores são todos ajudados, foi feito pela professora Débora, então eu queria agradecer no departamento dos convites para te associar isso para a referada. E queria convidar a professora Débora para fazer a apresentação do professor. Yes.
I don't understand, but it sounds so nice.
But it's the same thing that I said during the meeting. Eu preparei uma apresentação em inglês por causa do congresso, mas eu vou falar em português. É mais fácil porque eu não preciso ler nada. Então, o professor GABAergic é um colega com quem eu comecei a estabelecer uma colaboração em 2018, através de um edital da FAPESP para colaborações internacionais. Nós nós submetemos esse edital e foi muito bem sucedido. E a partir daí, nós estamos colaborando com outros auxílios da FAPESP e já estamos coletando alguns dados bem interessantes. O professor Gal tem especialidade em neurociência do comportamento e ele recebeu o grau de doutor em 1992 pelo Instituto Weizmann, em Israel, e também fez um pós-doutorado em Londres, trabalhando com o descobridor, né, o cientista que descobriu a LTP, Long-Term Potentiation, que é um índice neurofisiológico de memória. Então, os primeiros trabalhos do professor Richard foram com memória, ele ainda trabalha com isso, mas agora associado também a operações de comportamento emocional. E foi ele quem elaborou o modelo de trauma por imersão em água, que mimetiza um evento de afogamento em seres humanos. Ele atualmente é diretor da Faculdade de Psicologia da Universidade de Haifa, mas também já foi diretor da Faculdade de Ciências Naturais. Foi presidente da Sociedade Israelense para Psiquiatria Biológica e presidente da Sociedade Israelense de Neurociência. Tem tantos créditos aqui, eu preciso escolher alguns. Ele faz parte também do comitê consultor do Instituto Nacional para a Psicobiologia e é membro do Conselho Científico Grã-Bretanha-Israel. Ele publicou mais de 180 trabalhos científicos, tem um índice H de 70, foi orientador de mais de 60 estudantes de graduação e pós-doutorado, de pós-graduação e doutorado, e em 2013 ele recebeu um prêmio da Associação Israelense para Psiquiatria Biológica pelo conjunto da obra como mentor de cientistas, né, no início da carreira. Então, com esses pequenos atributos, né, que eu trouxe, que eu fiz uma boa filtragem antes, gostaria to invite you to give your talk. Thank you so much for being here. It's a pleasure to have you in our department, not only in the meeting, but also here. And I hope the students enjoy this opportunity, to meet a great scientist like yourself.
Thank you very much. I will not disappoint you. So I would just like to say at the beginning, first of all, thank you for the opportunity. And I'm not sure how many of you were also in the meeting that we were, but in the meeting I was talking about the main direction of our research, which is individual variability and how we work with this individual variability in response to stress and trauma, and out of respect to Goro's all the talks that I presented there. I'm not going to talk about it now, but you are invited to ask me about or talk to me about it later. Instead, I want to I want to take you through a journey of research that I've been participating in the last 20 years or so. And I hope that what will be clear is how this evolved over time and how the questions evolved over time, which I think is quite nice. And this relates to GABAergic interneurons and their involvement in stress and trauma and resilience. And the journey starts with the dentate gyrus, which is a region of the hippocampus, sub-region of the hippocampus that I've been working on. And actually, for quite a few years, a bit frustrated, because the dentate gyrus was not the most important part of the hippocampus. So much so that you need to explain why working on the dentate gyrus, because the hippocampus has been an area that has been really intensively studied, but mainly the C1, C2, C3 areas of the hippocampus, because I guess of the place cells, which are very impressive, and have led to the idea that the hippocampus serves as a kind of a cognitive map, and led to John O'Keefe receiving the Nobel Prize for these findings. And the dentate gyrus was always presented as a gateway for information to the real hippocampus. This started to change a little bit, not because of what we did, but because a lot of research started to realize that there was neurogenesis in the dentate gyrus. And I call your attention, you would find many papers that talk about neurogenesis in the hippocampus. It's not in the hippocampus, it's in the dentate gyrus. And one of the questions is, why does the hippocampus, sub-region of the hippocampus, has this unique ability, this unique feature, which, by the way, is a question that is not clear until today. But we came to that from a very different angle, to the focusing on the gyrus from a very different angle. And this angle is thinking about what we call emotional tagging. Emotional tagging is the idea that you need the emotional significance of the experience in order to memorize things. Because as every memory system, the hippocampus receives a lot of information, most of it not so relevant. This is not so relevant. Some of it is relevant, are things that you want to remember and so on. Some things are things that you better not forget. And the question is, what helps the system to identify those elements that they need to remember? And there are several mechanisms that could support. One of them is the emotional significance of the experience. And this is what we meant when we started to think about the emotional tagging. That if you think about emotional tagging, in behavioral neuroscience test, it means that you want to understand the mechanisms that are behind it, not just the concept. And how can it work? So one thing is, of course, if you look at, you want to study a memory-related system in hippocampus is, of course, a very good example because it is involved in memory, because actually it receives a lot of information, many of it, much of the information is not so relevant. It needs to be able to do exactly this kind of sorting. And it is involved in actually the consolidation of the relevant part into long-term memory. So it's a perfect region to study. And when you think about who can bring the emotional load, the emotional significance, you think about brain regions that are involved in processing emotional aspects. And the amygdala, of course, is one such region. And it has been known to affect memory processes and so on. So the idea we had was to ask whether activity in the amygdala can affect memory processes in the hippocampus. This is a translation of the question of emotional tagging to a mechanism. But what would you look at as a memory process in the hippocampus? We look at the proxy. This is long-term potentiation of LTP, which is considered to be a proxy of a memory process. And so the question became much more specific with priming the amygdala, stimulating the amygdala prior to inducing LTP, affect the level of LTP you can see in the hippocampus. And the idea is very simple then. You put the stimulating electrode in the amygdala, you set to record the LTP in the hippocampus, and then before you start the high-frequency stimulation to induce LTP in the hippocampus, you stimulate the amygdala in a certain way, and you see what the effect is. When we looked at CA1 area of the hippocampus, we found that priming the amygdala is effective. It reduces the level of LTP that you can see. However, this is very monotonous effect. Regardless of the different lines are different protocols of stimulation of the amygdala. The stimulation to the hippocampus is always the same. And you see that different protocols would lead to suppression of LTP. This is not the picture in the dentate gyrus. In the dentate gyrus, it really depended on the protocol used in the amygdala. So different protocols would lead to either enhancement or suppression in the LTP that you could get in the dentate gyrus. And this suggests an interesting thing. It suggests that the hippocampus is sensitive to amygdala priming. By the way, the same picture was also done exposing to different stressors. So the hippocampus is sensitive to stress, plasticity in the hippocampus is sensitive to stress and to priming of the amygdala as a mediator of some of the effects of stress. But there's a difference. The C1 area of the hippocampus is sensitive to stress, but not to what the stress is. It's sensitive to priming of the amygdala, but not to what exactly the parameters of this priming. Whereas the dentate gyrus is not only sensitive to stress, it is sensitive to the characteristics of the experience. And this puts the dentate gyrus in a very different and much more important position as defining what the hippocampus would be doing under different level of emotionality, stress, trauma. And this is where we started to be. Actually, it's not true. We were interested in the dentate gyrus to start with, but this is why we started in this direction. Okay. So the idea was, what if we change the dentate gyrus in some way? Would it affect the ability of the animals to cope with stress and so on? And to relate to some of the work that I did, I want to mention also the juvenile stress model, This is a model that we developed to study the impact of childhood adversities on the behavior or the coping abilities of the animals later in life, with the idea that the animals are exposed during a period that is resembling childhood in several aspects of their life, and then they are left to grow normally, and we come back to them in adulthood, and the question is how would they be able to cope with some stressful or traumatic experiences, with the expectation that this previous history would affect their performance. And we did find effects like that, and in this case, exposing the animals to the juvenile stress and then to the stress in adulthood resulted in a very specific, the other way around, a very specific effect on what we call local circuit activity in the dentate gyrus. Local circuit, very briefly, is a kind of description of the interaction between inhibitory interneurons and principal cells in the region, and you have different types of interactions. Okay. Because you have different types of interneurons, you have different types of such interactions, and we've been looking at electrophysiological protocols that emphasize different aspects of that circuit, and we found that this prior stress did not affect this protocol, that protocol. It's not really important what these protocols are at the moment, but you can see that there was a specific effect on a specific protocol, and this specific effect on a specific protocol actually suggests that specific interneurons are affected. It's not that all interneurons, not that all GABAergic modulation is affected. Specific ones are affected. And then we try to affect specific interactions within the dentate gyrus and see the effect. With one such interaction that I will explain in a moment, we didn't see much of an effect. With another that I will explain in a moment, we did see exactly the same feature. So no effect on this protocol, no effect on this protocol, but a specific effect on this protocol. Suggesting that we hit it, and this is without the juvenile stress. This is, instead of juvenile stress, we were kind of altering activity within the dentate gyrus, which was a very nice result. We were very happy with it, but what was even nicer, and I think more important, was the fact that this same manipulation also affected the behavior or the coping ability of the animals. So it was sufficient to have relatively mild modulation of activity within specifically the dentate gyrus to have a different ability of the animals to cope with the two-way shuttle avoidance task, which is a learning under stress task. And the animals were impaired in that, just as we previously found that animals who were exposed to juvenile stress were impaired in this specific task. So it seems that altering the functioning of the dentate gyrus, not just altering the function, altering the interaction of interneurons and principal cells within the dentate gyrus in a very specific way was sufficient to change the ability of the animal to cope. So this was like the first step. The dentate gyrus seems to be holding a critical role when you start to increase emotionality, stress, and more regarding what the hippocampus can do. And now, the next step, which was actually involved in the first step, as you will see, was trying to develop tools to work with specific interneurons in a rat. So we were interested in GABA, we were interested in interneurons, and it is not easy to study because you have different types of interneurons, and it's not easy to be able to manipulate specific interneurons and see their contribution. It has been suggested by some evidence that different interneurons may have different contributions, and some would be more involved in psychiatric disorders than others, and so on. But the tools are lacking. A lot of tools evolved working with mice that had kind of specific markers or pre-markers, for example, in specific cells or something like that, that you could target specific, also specific interneurons. PV interneurons is very famous for that. By the way, PV interneurons are not the only interneurons. It's like the first line of mice that could be affecting specifically the PV neurons, which led to a huge amount of papers about PV interneurons, but other interneurons are also important. Just that the tools will come later. But there is a challenge working with inbred lines of mice. And this challenge is that the inbred lines of mice, which are like genetically homogeneous, right? You would expect that because they have this kind of homogeneous genetic background, the level of variability, physiological variability, behavioral variability in these lines would be much less than in outbred lines. And this is one example to show that this is not the case. There are things that affect their behavior beyond the genetic background and so on. And one problem with the inbred mice is that you may have a very clear finding in one line, which does not repeat itself in another line. And this led researchers to emphasize the importance of actually testing findings. Inbred lines are important, but to also test the results and the findings in outbred lines. And since there are not many outbred lines that are still available, people are not working so much with outbred mice. Rats are mostly outbred lines. And it became more important to also develop tools to work with rats and test that. In addition to the fact that some of the behavioral referred to our rats as kind of higher face validity, resembles more what we know in humans than what the mice are doing. So we wanted to find a way to also target specific interneurons in the rat, despite the fact that you cannot do the same genetic manipulations. And here, I was very fortunate to meet Hans-Jürgen Wolfmer, a colleague of mine for many years from Germany. I actually met him in the context of both of us participating in European consortium that was on cell adhesion molecules, and both of us were kind of talking at the line, at the last rows of the chairs when there was a meeting, and we became friends, but also in their group, they were working on something that turned out to be very useful. They found that GABAergic synapses, in order to be functional, have to cluster in a certain way. And they need a molecule called gefferin as an anchor. The gefferin builds a kind of a network to which these actually the synapses are encoded, and it is required for the functioning of the GABAergic synapse. He also found that this gefferin does not work by itself. This protein works with some collaborating proteins that are important. And most importantly, they found that there are different molecules that interact with gefferin in different subcompartments of the cells. So that, for example, here in the axon, specifically in the axon initial segment, it is neurophasic. In the proximal dendrites, it's afferin A7, or track B. that you see interacting with Jeffrey. This means that if you would affect the expression of either neurophasin or FRNA7, you would affect the interaction of different subpopulations of interneurons with the granule cells. You do not affect the interneurons themselves, but you interact their ability to interact with the cells. So if you reduce the expression of FRNA7, you affect the proximal dendrite synapses, and if you affect neurophasin expression, you affect the interaction at the axon initial segment. And these are different cells, different interneurons that are involved. So we decided to use that in order to examine at least this kind of separation, the proximal dendrite interneurons and the axon initial segment interneurons. Okay? So this was the second step. We kind of had a tool to differentiate between the contribution of different interneurons. And I started to play with this tool in every direction we put it. So one of the things was to see whether these, as I told you, we are very much interested in the individual variability response to stress, where different interneurons contribute, which is kind of the individual variability. And here I need to mention another term. You know, plasticity, LTP is a form of plasticity. This is Tim Bliss, who was the person who identified long-term potentiation. I was fortunate enough to be posted in his lab. And some years later, Cliff Egron and Mark Baer came up with the expression of metaplasticity, the phenomenon of metaplasticity. The idea of metaplasticity is that if this is the level of plasticity that you can achieve without any intervention, if you do something prior to your attempt to induce LTP, you actually affect the level of LTP that you will get. So that the change that you make in this prior activation, which is actually plasticity, but not the plasticity you are focusing on, affects the plasticity that you will be focusing on later on. So as I said, this is a plasticity of synaptic plasticity. This is the changing of the probability of getting a certain level of plasticity. And I've been interacting with Cliff Egron for quite some time to expand this notion from really very narrow electrophysiological point of view to talking about behavioral metaplasticity. That is, for example, if you stress the animal and then you try to induce LTP, you change the level of LTP. It's a form of metaplasticity. And if you stress the animals and then the animals do not learn as well or learn differently, it's also a form of behavioral metaplasticity. And together we came up with this idea of behavioral metaplasticity. So we wanted to see if changing the interaction between inhibitory interneurons and principal cells would induce kind of metaplasticity. Okay, so started to focus on the amygdala, and the idea was to induce metaplasticity, long-term changes in the amygdala, and see how this changes the behavior of the animal. And to make things simpler, I will focus on two behavioral tests. We did more, but this would be good to demonstrate the elevated plasmase, which is indicative of levels of anxiety of the animal, and extinction learning, which is a learning that also is related to the ability of the animals to cope with the world, the changing world. That is, a world that was fragile and now is not. The stimulus is no longer actually marking a gain. And this is very important learning because it means that if the animal does not have extinction learning, they waste a lot of energy avoiding stimuli that actually no longer risk them. So what we did was very simple. We used one virus or the other virus to inhibit the one connection or the other connection of the critical cells with the interneuron. So here we started with the neurophasing, the action initial segment modulation, and we showed no effect on anxiety behavior, but a significant impairment of the extinction learning. On the other hand, affecting the other GABAergic synapses at the proximal dendrite did increase anxiety, did not affect the extinction learning. Now, this finding has several implications. First of all, it shows that, contrary to what was believed, the impairment in extinction learning does not necessarily result from changes in anxiety, because this was the way people were thinking about it. Increased anxiety, impaired extinction learning. These are two processes. More than that, the amygdala is involved in a different way in these two processes. You could separate mechanistically between the involvement of the amygdala in anxiety and the involvement of the amygdala in the extinction learning, which is again against what was believed. So the next step is this kind of starting to understand that within the amygdala you may have different behaviors that are being controlled using different mechanisms. I started with talking about the way we primed the amygdala and looked at the dentate gyrus. So, and injecting these viruses to the amygdala could, in principle, change the way the amygdala modulates other brain regions, right? And so we decided to look specifically into that. And for that method, we used the protocol that we studied before, where animals were exposed to, they were trained in the Morse water maze to learn a special learning task. And then they were exposed to the underwater trauma as a trauma. And about an hour later, they were tested again in this maze to see how this affected the performance. And also, in a similar way, the impact on LTP. So, and we found two effects. We found that this exposure to the underwater trauma impaired the LTP and impaired the special memory, as was evident in what we call the quadrant test. That is, you remove the platform and you explore how much the animal is exploring the area where the platform was. Okay? We used that, and now with the addition of manipulating the amygdala before. Okay? So it's the same thing, but now the amygdala is different, can be different. And what we found was that in both respects, manipulating the amygdala actually protected from those effects of the trauma. We did not block the activity of the amygdala. We only altered the very mildly the interaction between excitatory and inhibitory cells in this region, right? This region is there as functioning. They were more resilient, if you want, both in terms of the LTP and in terms of the special learning than animals that we did not manipulate. And this is really within the range of what you could expect even in normal population. This is not pathology. This is variability. Some would have a bit more of that effect of the synapses, some would have maybe less, and you get the difference in the co-capabilities. So this kind of relatively mild differences in the way the amygdala modulates activity and plasticity really seems to contribute to individual differences in the way these animals can cope with stress. Of course, if you take it to more severe manipulations, it could also be that it contributes to PTSD and so on and so forth. But it is also something that would work under normal conditions to kind of explain the differences between different people. So here we talk about the shifting from plasticity to metaplasticity because we modify the amygdala by the way we use viral vectors. In that respect is good because this is kind of, we change it not for the moment that we change it. It's a lasting change. It's mild, but it's a lasting change in the activity of this switch. And the result is that it's not only that the amygdala is modulated, but that the ability of the amygdala to modulate other brain regions changes. And slight changes like that of the sensitivity of the amygdala could lead to those individual differences. And now, I'll take another step from plasticity to metaplasticity to transregional metaplasticity. And it's again along the lines of what I showed you. So it started with the work that we did, reducing the expression of neurophacin and looking at the impact on different aspects of behavior. As I told you, this manipulation actually affected extinction learning. Now, because we modified the amygdala, we wanted to verify that indeed the amygdala was modified. One of the validation experiments that we did was to see if this manipulation changed the ability of the amygdala to induce long-term potentiation in the prefrontal cortex, because we knew that primary or applying high-frequency stimulation to the ELA would lead to LTP in the prefrontal cortex. So the idea was whether this manipulation changed anything in this LTP, and indeed we found that LTP was impaired. Perfect. We can show that the virus did its job. Okay? And my supervisor, my PhD supervisor, told me once, when you have such a good result, stop. Because if you continue, you'll get into trouble. And I did not listen.
Like it was.
Now, this was way after I was a student of this, but what we wanted to do is to further verify the fact that it was the amygdala that was modified. And we said LTP in the DLA to prefrontal is affected. Good. LTP from another pathway that goes to the prefrontal cortex should not be affected. And this would be wonderful. We would show that this other LTP is functioning, the DLA LTP is not functioning. We will show that our manipulation worked well. Unfortunately, this LTP was also affected. We were very surprised by this, so we decided we need to repeat it. And this time, we thought maybe somehow stimulating the ventral subiculum, which is the pathway, but somehow activates the amygdala and goes. So we anesthetized, locally anesthetized the amygdala and did this experiment again. Now, without any possible involvement of the amygdala, instead, the LTP was unfortunately impaired. So we decided to examine behaviors that are suggested to be dependent on the prefrontal cortex, not on the amygdala. And one such task is reversal learning in the water maze. So the initial learning is known to be campus dependent, but reversal learning, that is to shift the platform to a new location, the animals have to run the new location, is considered to be very much dependent on the prefrontal cortex. The other aspects of the water maze learning were not affected. The initial location of the platform, both groups were as good, but when we repeated the behavioral quadrant test, you can see that these animals stick to the previous location of the quadrant. So reversal was not working. And furthermore, if we looked at the activation of the neurons in this region using sequence activation, we found a reduction in the BLA, which is expected. This is where we manipulate. But we also found a reduction in the prefrontal cortex, which we did not manipulate. This means that there was something that was transferred as an alteration to the medial prefrontal cortex without us doing anything, except letting the animals live. Okay, it's not an immediate. So here we go a step further. So we have plasticity, we have metaplasticity, which I remind you is the fact that the amygdala was modified, changes actually its influence on other brain regions. But now we talk about changes that are independent of the that induce some, the amygdala probably induced some change because this was the only thing that we had manipulated in these animals. But now the change that occurred probably is a kind of a result of the different interaction between the regions. Induces some form of plasticity in the prefrontal cortex, which induces a difference in the way the animals go. I remind you that one of the questions we had was contribution to individual variability. You can see that if two people have slightly different amygdala, in the process of time, they would also have slightly different prefrontal cortex, etc. So, to summarize what I was saying, we talked about the dead age hours as being important. And I can tell you this was a relief for me because I was working for many years on the dead age hours, apologizing for not working in the single one. So now I can say, okay, the dead age hours is important. We, on this journey, were able to develop a very useful tool to kind of differentiate between contribution of different subpopulations of the interneurons, and then to use that tool to actually start learning about the differences in mechanisms that affect and the way that maybe individual variability works. But also, on the way, we could identify actually a different dissociation between functioning of the amygdala that were not known before. And we also found that by changing the amygdala, you have to change also the way the amygdala modulates other brain regions, so the whole interaction of the amygdala with other brain areas can be different, and it has behavioral consequences. And finally, whenever we do a very local manipulation, we're very proud of the localistic effect, we have to check that we really, I would make it easy for some of you. When you do a pharmacological manipulation, it's a short-lived manipulation, probably you are. But when you do viral vector exchanges, when you induce really this metaplasticity effect, it would have additional consequences as time goes by. And you need to control for that, or test that, or learn what it does. And the main thing, the journey. The journey was really a fantastic experience, and an experience of working together with different collaborators, working together with different students, which we always actually know that they do work. And so I thank my students. This is like the current group I have. Collaborators, of course, money. And thank you very much.
Thank you very much, Professor Gall, for the wonderful lecture. Really interesting questions. I think we can open up for questions.
I have a question. So at the beginning of your talk, you were saying that you expose animals to stressors, and that also induce changes in how the family gyrus was functioning. Did you test what were the nature of the stressors? Did you test only neurogenic stressors or also physical stressors, mixed stressors?
The comment on the different stressors is not only our work. There were, since '87, I think, there are papers that talk about the effect of stress on LTP and C1. I think Thompson was the first one. Many studies who looked at the impact of different range of stressors on LTP and C1.
And it's regardless of the nature.
And then it's always suppressing LTP and C1. This is very common and a lot of loss. There are much fewer studies of different laboratories that look at the effect of stress on the naked gyrus because it was less important to study. But there are several such studies, including ours. And then actually it's a range of stressors. It could be more physical, more chronic, more aggressive, and so on. It really, looking at the differences, it covers different variables that contribute to the experience of stress. What we have not done, we started with the stress, and then we moved to actually looking at the amygdala priming. And with the amygdala priming, we did a systematic test. We did not come back to test different stress protocols in a systematic way. So I cannot tell you which stressors would do what, but the fact that you have a range of steps that could result in a different outcome, it's not only our work, it's something that has been published. And I know more about the protocols of priming the amygdala and what they would do than about the exact nature of the stress.
And the effect is intensity sensitive, or I mean, if the stressor is more intense, the reduction in LTP is more intense. Is there a dose response?
We did. The dose responses we did was with priming the amygdala. We did not do that exact systematic studies with the stressors.
Okay.
So I cannot really answer that. My feeling is that it is not just the dose or the intensity. It's very difficult actually to define the intensity of a stress experience. We often, when we describe the stressor, we think we describe the stress experience, but it's not the case.
Right.
The stress experience is something subjective in the individual. I think that the parameters of the amygdala are more subjective in a way, because it's the way the stress would activate the amygdala.
Yeah.
So, as I said, unfortunately, I don't know the answer about what parameters of stress would lead to. I know that different stressors would lead to different results. And we did systematic studies with the amygdala priming. It's not always the dose. It could be the frequency, it could be the pattern that could affect, which actually reflects the way the amygdala processes the experience.
That's so interesting.
Thank you.
It's amazing.
More questions?
So, maybe one. Do you guys looked brain oscillations, like amygdala oscillations, prefrontal cortex oscillations, because one of the things that interneurons are doing is to modulate oscillations, to create oscillations. So I was thinking that can be a mechanism behind the data. And also, I was wondering, do you know if the BLA projections to the prefrontal cortex are the same synapses or neurons that the projections of the ventral hippocampus? Because this can explain some modulations, but I'm thinking you don't have any change in the initial evoked potential, just on the speed, right?
So these are several questions. First of all, an interesting question about the effects on frequencies. I've been collaborating with Oliver Stork from Amygdalar for similar line of work. And he has been actually starting his career working with Hans Christian Pappe, who was one of the people who looked at the oscillations and the effect. So this was something that we have discussed quite often, but also looked a little bit at this, but the manipulations that we did do change some of the frequencies in the amygdala, and they become synchronized the waves, the oscillations. So it could be that this is part of the effect. In fact, we are now submitting a proposal to the FDA, looking at more at oscillations, at a map of pulse-emitting patterns of activation, and so on, using similar manipulations, so that we hope we really have maybe a more informative answer to that. But it's true that different studies looked at either the level of oscillation or the level of interneurons and so on, have close relations. Yeah. However, one thing that needs to be said, when I started being interested in interneurons and plasticity in interneurons, there was a very strong and quite aggressive group of researchers who were standing against the possibility that there is plasticity in interneurons, because they are like the pacemakers of the brain, and it would kind of change the time measurement of the brain in a way. And so these were people who were in very influential positions, and they resisted any indication of plasticity. Only in the middle of 2000 something, one of them is Thomas Freund, started to say, okay, maybe some interneurons are responsible for the frequency and time, and some are responsible for modulation of other aspects. Mm-hmm. And I think that this is more in the direction of what happens. So not all the interneurons, the main real function of the interneurons is about oscillations and timing. And there are some who are much more influenced by neuromodulators and like serotonin, dopamine, and cannabis and so on. And they probably are not the ones that are contributing to the timing of the effect, but actually to modulation of activity of the cells, which contribute to behaviors such as we have shown here. Nowadays, at least, it's possible to say that there is plasticity in interneurons, but you need to verify which interneurons and so on and so forth. So this is about oscillations. Mm-hmm. And hopefully, if we get the grant, we will have more information about that in some years' time. I'm trying to remember the
The question was about the projections from the BLA and the ventral hippocampus to the cortex.
So, I should remember that because this touches upon a very important point. All the electrophysiological recordings that I've shown you in the C1 and the dentate are in the dorsal dentate gyrus and the dorsal hippocampus. This is because technically you can record field potentials in the dorsal dentate gyrus. I don't think that anyone knows to record field potentials in vivo in the ventral dentate gyrus. The BLA does not project categorically, does not project to the dorsal dentate gyrus. So this is not a direct manipulation. The main concept in the field is that it does project to the medial entorhinal cortex, which affects the dentate gyrus, or to the medial prefrontal cortex, which can affect the activity. We actually recently have new data showing the ventral hippocampus, the ventral dentate gyrus, which is affected by the BLA, can modulate plasticity in the dorsal dentate gyrus. We have very nice results of that. So what we did was to have electrodes, just as we had in the BLA, electrodes in the ventral dentate gyrus, trying to affect LTP in the dorsal dentate gyrus. And actually, we do see very nice modulation. So it could be that this is also a pathway of BLA affecting ventral, affecting dorsal hippocampus. And again, this is another application that we are trying to submit now with these results. That I hope to have more information about in coming years.
Okay. Thank you.
Your next visit.
Okay, thank you very much.
Thank you very much. Thank you very much. Obrigado, pessoal. O que teria a transmissão agora? E vamos lá. O próximo. É, eu não vou fazer entrevista. É, mas precisa fazer.
Não vai fazer entrevista.